Hey, we're back talking about a couple different topics here. I wanted to at least get a little section to introduce cell signaling. This is cells' methodology of receiving environmental cues. Okay, there's a lot of variation in cell signaling. I'm going to talk about lipid-based signaling, but there's also protein-based signaling. And if you see the little cartoon there, isn't this a neat cartoon? So the hormone, the neurotransmitter, binds to the receptor. There's a conformation change, and another protein binds and becomes active, and a lot of times it's an enzyme, and it then further activates a cascade of proteins to transduce the signal. So a cell converts one type of signal or stimulus into another. The ligand binds the receptor. An enzyme binds to the receptor. It changes conformation. The enzyme is active, and it does its thing. It interacts. And this is just a cartoon showing kind of a protein-based signaling. So that's one. There's a lot of different variations of receptors and signals and different tissues, different conditions, lots of differences. So I just wanted to kind of do a callback to the Lipid-Ralph models. And not only is people try to figure out the structure of a Lipid-Ralph, but how does a Lipid-Ralph organize, or how are the receptors, whatnot, organized in a Lipid-Ralph? So single Lipid-Ralph models here, receptors are associated a steady state, and it can be activated, and in this case dimerization occurs. Antibodies, ligands, etc. Dimer causes a dimerization, and then there are other molecules, signaling molecules, recruited to this side of the transmembrane receptor. And so here you have the Lipid-Ralphs, the receptors inside the Lipid-Ralphs. So they're in a Ralph. Maybe the receptors are outside the Ralph, and when there's a signal, then the receptors come into a Lipid-Ralph and are kind of forced together. There's a different fluidity there in a Lipid-Ralph. And so they may even have weak affinity, but they could dimerize or become oligomers on lipid binding, and that can lead them to being retained on a Ralph. So there's two kind of flavors right now of how receptors and lipid Ralphs, there's two flavors of the single Ralph. Then there's the clustered Ralph models that says that different receptors are in different Ralphs, which themselves are different. And it shows the extracellular, the transmembrane, and the cytosolic elements here. So receptors are activated. They could cross-link proteins to other proteins within the Ralph, and maybe the Ralphs collapse together, and you have a collection of signaling going on. And so the Ralphs that are separate actually are folding together, bringing in the receptors, and you're getting a massive effect based on some type of signals that may be occurring there. Clustered versus a single. Clustering can occur extracellularly or in the cytosol, so they're saying organelles. And these models are not exclusive. That means that it's very possible the cell does both models. Here's our GPI anchored proteins that could be the primary, or after the activation could continue to stimulate the whole response. And it doesn't mean that both models can't be part of how cells deal with signaling as far as through lipid raft. So here's a cartoon. This is an insulin growth factor, insulin receptor. And so when those receptors bind to insulin, an insulin response element here binds to the receptor, the cytoplasmic end. And then this molecule can activate one pathway or maybe another pathway, maybe both, maybe other pathways. And you end up getting signaling to grow, divide, to survive, or you could get death signals as well. And that's called a signal relay process here. So when you did a protein interaction material technologies, this would only interact with these enzymes when it itself was bound and activated. So it's sometimes a little hard to make these connections here. But FRET and other kinds of techniques allow you to see recruitment of these complexes to the plasma membrane and start putting together the pathway, as it were. And you can measure the genes that are coming on in the nucleus in response to some type of stimulus, such as insulin here. So besides proteins, there are also lipids that can be modified, and they themselves cause the signal. So in this cartoon, number A, here's some type of receptor bound to a ligand, and that activates, and that's what this little bars are showing, like a little sunshine here, and that then targets or binds to an enzyme known as phosphatidylinositol 3 kinase. So if you remember our lipids, inositols are part of phosphodienosyls are part of lipids. And so what happens is this enzyme will find a phospholipid as its substrate. It will phosphorylate the lipid, and that phosphorylated lipid recognized by a signal protein that then might be an enzyme or might dock with an enzyme, and you get signals to occur from that direction. So there's a kinase signaling, so where the lipid is actually phosphorylated, and then this protein binds to phosphorylated lipids. Okay. B is called phospholipases, generate signaling fragments. So in this case, instead of a phosphorylation, the receptor ties into a phospholipase, in this case phospholipase C, and its substrate is this same phospholipid, but this time the phospholipid is broken, and the IP3, this little red little candy here, is known as IP3, inositol phosphate triphosphate, and this fatty acid is diacylglycerol, DAG. So this phospholipase breaks apart the lipid into a DAG molecule and an IP3. Turns out that the DAG molecule can do signaling, and this IP3 actually will go to the ER, and you'll have a calcium-based signaling there. So this is uh, two paths. So you have one signal, but you got two different relay systems that have been activated here. So a phosphorylation or a lipase that breaks apart the lipids. And the secondary molecules are said to be second messengers. So the primary growth factor hormone, that's the primary messenger, first messenger. The second is through the relay system. And here's some examples. Okay, 5 different second messengers. Sometimes a receptor causes the formation of cyclic AMP or cyclic GMP, which then activate other pathways. Calcium can be released in the cell, and that causes signals. Here's our DAG, diacylglycerol, and that activates protein kinase C. So DAG activates protein kinase C. A protein kinase is an enzyme that phosphorylates targets. So this activates a kinase that then activates its targets. And eventually you're getting probably turning genes on or turning genes off, right? Here's our inositol triphosphate, IP3 is released, and it actually opens a calcium channel and releases calcium that causes changes in the cell. And PI3 phosphates are that's with the kinase. Once they're phosphorylated, then that can serve as a docking platform for other signaling proteins. So there's a lot of different ways, versions of how the cell does signaling. You know, the question is, why not just do one or two? Why have all these complicated mechanisms? I think it's tissue specific, stimulus specific, gives more flexibility. You can fine tune a response if you have multiple pathways tweaking on and off there that can give a mediated response based on the true nature of the environment the cell is facing. Here's just a kinase here will phosphorylate the target here, and it can work with protein kinase B and AKT, which is another signaling molecule. Lipase gives us the IP3, and you're getting the calcium channel, or the DAG is protein kinase C. Yes, there is a protein kinase A. We're just not talking about that. So membrane phospholipids are used, okay, as secondary messengers. They're PIP2, that's a diphosphate, phosphatidylinositol diphosphate, can be split to IP3 and DAG, or can be phosphorylated, depending on the enzyme that's activated, and you end up getting a kinase here or there potentially Gunnar-Gose reaction. Here's just showing a cartoon showing the more chemical structure of the DAG, protein kinase C, and the calcium pump, releasing calcium from endoplasmic reticulum. Inositol 1,4,5 triphosphate IP3. Chemists name these carbons, carbon 1 to what is it? 5. So this is 1. What is this? 4. So it's 1, 2, 3, 4, 5. So it goes in a counterclockwise position. So that's why it's called 1, 4, 5 triphosphate. So the overall picture of lipid signaling is shown here. Here's a PI phosphatidyl diphosphate. The lipase cuts it to the DAG, which then actually interacts with protein kinase C, and you end up getting substrates that leads to a signal. Or you have IP3 that binds to sensitive channel, this binds and it opens up a membrane channel, so it changes the shape, the conformation, and you have a flow of calcium out. And some of that calcium binds to protein kinase C and changes its conformation such that it can bind to the DAG and causes the phosphorylation. Or sometimes material from outside the cell causes calcium influx, and you can activate that pathway as well. Or the calcium can be released outside the cell. So there's a lot of different things that can happen here, but it all starts with a phospholipase being active. Okay, and certainly this has to be inactive, right? You can't have an active phospholipase all the time, or you just keep chewing all your lipids up. So it's on or off. There has to be kind of an on or off switch there. Okay, so that's just a simple cartoon series that talks about phosphorylation a little bit and lipid signaling using signal transduction using lipids. There's also proteins with associated with other second messengers, but I just gave you a quick example of the one here. Okay, let me journey a little bit into membrane proteins. So we've talked a lot about receptors and membrane proteins, and this is probably, you saw this from high school biology. Here's the phospholipids, the steroids, the cholesterols, here's the actin are bound here to proteins that are peripheral or transmembrane through. Some of these are bound with carbohydrates, glycolipids, and there may be outside the cell fluid and other kinds of matrix that hold cells into what we call a tissue, a liver, a kidney. So there's a lot of anchoring going on. And of course, cancer cells have abnormal structure, and so they tend not to be so anchored, and then they tend to move around, and that's not good. So fluid mosaic, integral membrane proteins are transmembrane, and then those that are peripheral bound to the surface. Membrane proteins are involved in transport. Some ions are taken in or move out. Some are passively moved in. Some you may have to use energy to change the conformation and move material out. Notice the concentration curve, a lot of blue out here, a little bit of blue here, a little bit of red, but to go against the concentration gradient, you have to use the energy to pump it back out. Membrane proteins can be enzymes, okay, that they can interact and have a substrate and another substrate, and you end up with a product here. Signaling lipids, and you've got now an activated receptor. Two cells can bind and actually talk to each other by membrane proteins. Here they are interacting. Some cells can join very tightly. Our epithelial cells are bound very tightly. And again, they can bind to matrix molecules outside the cell to make the tissue or the organ. Okay, so they want to stay in place there for their activity. Carbohydrates can be attached to the proteins or lipids that are in our membranes here. Okay, and they help in cell recognition, these glycoproteins and lipids. And if you're doing the transplant, part of the rejection of transplanted organs and whatnot is because people have different types of carbohydrates and glycolipids, and your immune system understands self, but now you've got something that seems to be non-self, and that's an issue. Where are the lipids made? They are made in the ER, and really in the smooth ER. There's no ribosomes there, and there are vesicles that go through our cis-medial trans, and then through constitutive here they move receptors and lipids back to the plasma membrane. I think we talked a little bit about that. And so membranes, if you're going to purify them, you have to isolate them, usually in the presence of detergent. So you have to do empirical figuring out how much detergent to add to your mix to try to get membrane proteins to stay soluble. And you change the ionic strength, the salt levels, a lot of different processes to purify membrane proteins. A lot of times you have to have detergent. Okay, so membrane proteins can have areas that span the lipid. The first glimpse of a membrane alpha helices was by an image of a bacterial rhodopsin in 1975, and that experiment then allowed us to predict. So the first protein informatics was starting by predicting structure here, and they developed a hydrophobicity scale, and there's software that will track the amino acids based on how hydrophobic or hydrophilic they are, and you get this little scale here, and it shows all these different regions of the protein that are going through the lipids here. So you can figure out, here's from the first amino acid till the end, you can see how many lipid spanning regions there are through the plasma membrane here. So you can make predictions. And like I said, there's scoring mechanisms where they'll give a certain score to be hydrophilic, hydrophobic, and you can figure that out. They started looking at these coiled structures, but they found here's a beta barrel structure with pleated sheets that makes kind of a round with a hollow structure. Materials can go in and out. Complicated barrel structure. So a lot of the structural biochemists, this is what they do for fun, is try to make models to understand the flexibility and the nature of the function of these pores and receptors here. Here's a potassium ion channel looking down, and turned out in the middle there was a mechanism to where only the right amount of material could come through into the cytosol, the bacteria. They don't want everything to come through, so they changed the amino acids sitting out, and that allowed whatever needed to be taken in, attached, and carries through the pore. Here's a transmembrane with an alpha helix. Here's multiple, 3 alpha helix. Here's a barrel structure. Here's an amphipathetic helix here that attaches to a membrane. So the amphipathetic, the charges are on one side, okay, of the helix there. Here's our GPI anchorin that we've talked about, and these can interact, and they might even be clipped once they have a successful interaction on either side of the lipids here, and you build kind of a machinery complex going. Here's 2 different proteins that are interacting. So think of all the technology we have to see if these 2 proteins are interacting with each other. Here's the protein anchored by a fatty acid, different types of fatty acid anchors and structures. And again, you wonder why not just do it with one. So I think there's a lot of fine tuning and probably some secrets here to the structure. We won't get into this is stuff in biochemistry you'll have to memorize and learn. And again, the bioinformatics, your personalized medicine will be able to predict how good your body is at making these membrane proteins and the glycosylations and the GPI anchoring, and maybe there's some association with diseases and whatnot. So this shows an alpha helix through here and the hydrophobic core. About 20 amino acids are required to go through a lipid bilayer, so you can make predictions. Here's a membrane protein glycophorin and hydropathic index on this side is hydrophobic, and on this side's hydrophilic. So by running the protein's amino acids, you would predict this is the domain that binds and goes through the lipid bilayer here. His bacterial rhodopsin, 1, 2, 3, 4, 5, 6, 7. I call these serpentine receptors. And you can see what parts of the protein are going to be in the fluid versus ones that are stuck in the lipids here. So you can model bacterial proteins and look for these structures here. And this shows proportion of predicted membrane proteins. We have a lot of membrane proteins, but so does yeast and E. coli. So we're not just such that we have so many membrane proteins. We don't have as much as you might think. It's just shown another structure, how if there's a binding, so this strand here, and here's a 12-stranded receptor, very close together in structure, but still different. So again, trying to figure out the nature, and here's just some more sophisticated barrel structures here. Here's the another transmembrane through this area. This has cytosol. This is, and notice the SH, the sulfurs are not linked, but outside the cell, it's an oxidizing environment, and you get then these disulfide bonds that cause this structure to be maintained. Here's the carbohydrates as well. So you can make predictions based on the nature of the protein and the environment as to what structure it will maintain. Again, membrane proteins have to be solubilized and usually purified with some type of detergent. And lipids stuck in will tend to form this structure, kind of a mycel structure. And here it is. And if you throw them in with detergent here, so you membrane protein lipid layer, if you add the lipids here, after you've kind of denatured, you've got a mycel formation. But if you add the detergent to the protein, so here's your lipids are forming, but the protein is coated by the detergent and it stays outside the lipids. So this is the lipids, this is the protein in the lipid, and when you add detergent, you get the lipids by themselves and the protein by themselves. So it's a way to separate out a protein from its lipids based on having a detergent. The detergent actually takes the place of the lipids and covers and binds the transmembrane region here. So that excludes the regular phospholipids, and they then tend to form aggregates amongst themselves. So this is how you can purify a protein with detergent. Here are two common types. We've talked about SDS. Here's another one called Triton X100. These are detergent molecules that take the place of phospholipids and coat the proteins here. And so mild detergents for solubilizing and then reconstructing in a kind of a cell-free system concept. There is the sodium potassium pump. It's two proteins inside the lipids. When they add detergent, notice the two pump dimers are purified. You purify them, you remove the detergent, and you add your own lipids, whatever you wanted, and notice you reconstructed, but this way you reconstructed the pump inside basically an artificial lipid bilayer, and then you can study what's coming in and out of the bilayer here, and you could test with temperature, poisons, chemicals to see what's driving the function of the sodium potassium pump. You can actually add material in and follow it in and out of the cell. So here we take the pump directly out of its natural membranes and we put it into an artificial membrane system. I mean, it's real lipids, but it's what we made, and now we can test the pump for different parameters. Really a neat concept here, kind of extending the cell-free system where you're actually making an artificial cell. So the synthetic biologists are already aware of this, actually making and decorating an artificial cell with things like a pump. Okay, I won't stop here. We're going to pick it up with studying the first really good way to study the lipids on either side was found in red blood cells. And this cartoon shows some of that process. So we'll pick it up next time going through red blood cells and continuing our journeys here on membranes and membrane structure and membrane proteins. Come back.